Good morning, everyone, and welcome to EAB University. And this webinar series is funded by the USDA Forest Service. My name is Robin Usborne, and I'm coming to you from Michigan State University. Along with my EAB University colleagues, Amy Stone from Ohio State, and Sarah Stack and Dr. Cliff Seda from Purdue, we welcome you to today's presentation by University of Massachusetts Professor Roy Van Driesche, entitled Biological Control of EAB, Putting It into Perspective. Dr. Van Driesche has a bachelor's degree in entomology from Oregon State University and received his PhD in entomology from Cornell University in 1975. In 1976, he joined the University of Massachusetts Amherst and worked 39 years in several capacities, including biological control of fruit and vegetable pests, greenhouse pests, forest pests, and pests of natural areas. Now retired, he's working part-time on biocontrol of EAB and writing a book on integrated biological control into conservation practice. Before we get started, I wanted to remind you that your comments and questions are welcome today. Please feel free to write them in the question and answer feature, which you can find by mousing over the bottom or occasionally the top of your screen. We will be making a note of all the questions, and Roy will be responding to them after the presentation so we can keep this webinar flowing smoothly. To keep these free webinars coming, we do need your feedback. There will be a link provided to you in the Q&A pod at the end of the webinar for a short, voluntary, confidential survey that we hope you'll take time to fill out. If you're one of the first 10 people to fill out the survey, we will be sending you an EAB goodie bag. But we hope you'll give us feedback either way. I will also be sending out an email after the webinar with that link in case you weren't able to access it during the webinar. For those of you wanting CEUs, if you would like a certificate indicating that you participated in today's live webinar, complete the survey and send an email message to Amy Stone at stone.91@osu.edu. Certificates will be emailed to you within a week of today's program. I also wanted to tell you that you will also find all the recordings for this webinar and all previous EAB University webinars at www.emeraldashborer.info. Thank you for attending today, and Roy, you can now unmute your microphone and share your screen with your presentation, and we'll get started.
Okay, Robin, can you hear me?
Sure can. Looks good.
All right. Well, I'm very happy to be here today and to be speaking to you about biological control of emerald ash borer. I've been working on this with a group of colleagues since 2008, and the work that I will discuss with you today really is rooted in the foreign exploration and founding work of Leah Bauer and Julie Gould, who is my clicker visible when I point to things? Anybody? Okay.
Yes, it is. Go ahead.
Did the initial foreign exploration work for finding natural enemies in Asia, and Jian Duan of the USDA ARS, who has led our evaluation work since 2008 in plots in Michigan, and more recently with the establishment of parallel plots in the northeastern United States under the direction of Joe Elkington. Joe has also joined our group. Well, I'm sure that most of you already know that EAB showed up in Detroit, Michigan in 2002, although it had probably been there for perhaps a decade, having arrived earlier in wooden packing material, probably from China, but that subsequent to 2002, it has spread rather steadily across the eastern United States, now reaching as far south as Louisiana and Georgia, and as far west as Colorado, and up to almost, if not in Maine, on the east side. Also including several provinces in Canada, but has not yet made it to many other areas of the United States that have potentially infestable ash species. Well, before we get into the focus of today's talk about biological control, one of the things that I've learned in my work recently with ecologists is that the question that needs to be asked before assuming that biological control is the answer to any particular invasive species problem is to consider carefully the question of what is the fundamental driver of ecological degradation in a system. And while in insects this may seem entirely straightforward, with invasive plants often it is not. For example, with garlic mustard, which was widely assumed to be a primary driver of degradation of forest understory herbaceous plants, it has now, through more research, been shown to be much more complicated and to be, to a large measure in many locations, rather a passenger that's pulled along by excessively high deer populations and invasive earthworms. But for emerald ash borer, it is abundantly clear that this is a primary driver of ecological degradation. We see high levels of tree mortality, as seen in this picture here, in relatively undisturbed forest stands along rivers with old growth ash trees. This was very typical of many of the sites we worked at in Michigan, as well as, of course, in more disturbed, more recent forest stands, as shown here, showing some dead ash trees along just a relatively recent forest development. And also, before making any assumptions that biological control is necessarily the solution for a particular problem, one has to think broadly about all the other possible ways to approach the problem. And in the case of emerald ash borer, one must also think somewhat differently between what we can do for forests and what we can do for urban shade trees. For urban shade trees, for example, there are options such as treatment of individual trees with various injectable pesticides that are quite effective for providing 2 or 3 years of protection, to replanting urban landscapes with exotic species of ash trees that have natural high levels of resistance, such as the various Asian species. However, for forests, those are not viable options. We might, however, wonder whether the same kind of approach that's currently being taken for species such as American elm and American chestnut, where resistant lines have been selected and now they are being replanted in wild areas, whether that would perhaps be an effective method. And that is being investigated by researchers such as Dan Herms and his colleagues in Ohio, and it may well lead to lines of trees that are useful in urban settings. Whether that can be done for forest is a little bit broader question that remains to be seen. Another complication, of course, is that we're not talking about a single tree, as in the case of chestnut or elm, but rather a suite of trees in the genus, 3 at least in the east that are strongly affected are white, green, and black, but there will be many others as this infestation spreads. The focus of today's talk will be principally on the remaining items on this list, and that is sort of the potential impact of the species of parasitoids that have been introduced from the native range in Asia and how they will interact with the native natural enemies that are already present and whatever level of tree resistance may exist in native North American ash. So just as a little further bit of background, it was attempted to eradicate emerald ash borer. This was quite a large effort on the part of APHIS, but because infestations are quite hard to detect because of the limited power of the traps that are available given the biology of the species, it has not been successful, and this approach was abandoned. We do know from common garden experiments that the North American ash trees generally are quite susceptible to emerald ash borer. All of the 4 groupings of trees to the right in this chart are mortality rates of respectively green, white, and a black ash by Asian hybrid, and they all were very susceptible to emerald ash borer, showing increasing mortality over time. In contrast to this first set of bars, which is an Asian ash that shows stable high levels of resistance. That said, our work in plots in Michigan have shown us that even American ash trees, while they become overwhelmed and die when attacked at high rates, do make an effort to kill young larvae, and that this level of kill is most effective when the attack rate is low and the trees are healthy. And we have seen something on the order of 10 or 20% of very young larvae being killed by the ash trees. So I framed the rest of this talk sort of around some commonly asked questions about the system. The first one listed here is whether the native parasitoids are likely to control emerald ash borer on their own. There are some parasitoids, particularly species in the genus Ecological, which is a type of raconid wasp, that are beautiful creatures with these lovely red abdomens that you see right here. And the level of parasitism of these species, while in the first 2 years of our study was almost undetectable, quickly soared to very high levels of nearly 50%, only then to sag again to relatively modest levels of about 10%. Now, I must say that while this data shown here fits our general assumption about these native parasitoids, that they are somewhat generalist. They're moving over to EAB from other agrylus species like bronze birch borer in the forest, attacking a suitable and a highly available host when it is highly available, but not being able to track it as its density falls. That said, however, it may be a little more choppy than described by this rather smooth trend, because in 2015, which we just took our data in our plots in Michigan about 3 weeks ago, we saw very high levels again of Ecological parasitism. So I do feel this is a very important part of the natural enemy complex shaping what's going to happen to EAB. Another commonly asked question is the role of woodpeckers, and what is that now, and is it likely to change? And what we've seen in our plots over time is that woodpeckers have steadily caused a rather stable level of 30 to 50% mortality to the large larvae, which means the last feeding stage, the L4s, and the overwintering JL or pupal stages in their chamber. And over the period from 2008 right through, I can now say 2015, this seems to be very stable. As you can see in this data here, where the level of attack is high and is not really trending up or down. And I can say, even though we haven't tabulated yet, that our data from 2015 is probably higher than it was in 13 or 14, again, showing the stability and importance of woodpecker attack. The underlying assumption we have about woodpeckers is that they responded to a very abundant food source by reproducing, and that woodpeckers being somewhat long-lived, average lifespan of maybe 6 to 7 years, that effect will last even after EAB declines for a while, but that at lower densities, eventually the woodpecker population will decline as well. But again, I must stress that has not yet occurred, and woodpeckers remain one of the largest sources of mortality affecting EAB in our central Michigan plots to this day, and have done so now for 8 years. As a small side note, I want to address the question of whether these EAB parasitoids, whether the natural native ones or the introduced ones from Asia, could be used on a mass rear and release basis in a more or less biopesticide manner. We do now have considerable capacity to produce these things at the Brighton APHIS mass rearing laboratory, but the catch is that this is an expensive rearing system because of the need to manipulate logs and then to rear a long-lived species of host and then produce the parasites. So at $2 a parasite, these are suitable for infesting new areas, but they are not suitable for mass rearing and releasing the way one might do in greenhouses, an area that, as mentioned in the introduction, I have had some considerable experience. So the excuse me. The history of classical biological control began really with the leadership role that Leah Bauer and Julie Gould and others from USDA played in conjunction with their Chinese counterparts, Dr. Wan and Dr. Yan, in the period particularly of 2003 to 2007 or so. A series of parasitoids were identified in China, and of these, one was quickly identified, not shown here, as being too general to be considered as a biocontrol agent and was dropped for further consideration. But the remaining three looked promising and were brought into laboratory culture where their host ranges were evaluated. And this culminated in 2006 with a petition that was filed with APHIS for permission to release, which was granted for these three species in 2007. Major releases then began in 2009, was the Brighton lab geared up to make that possible. Foreign exploration then continued in Asia, but a little further east and north in the Russian Far East under the leadership Jin Duan of USDA ARS in Newark, Delaware. And he found three important species of parasitoids there. One of these, Theus galeni, became the focus of his work and it was similarly evaluated for safety and a petition filed with APHIS for its release, which was just granted in April of this year. And we're now gearing up with the Brighton lab to support mass releases of this parasitoid here. The other two species either were difficult to rear in the case of this Actinicollis species, or needed further study to define the entity better based on its genetics and reproductive biology, as in the case of a new egg parasitoid in the genus Oobius. So the three species from China were widely released during the last 7 to 8 years in many states through a rather extensive program sponsored by APHIS in cooperation with various state agencies and universities. And this has resulted in the successful establishment of two of the three species on a wide area. So the egg parasitoid Oobius agrile has established in a number of states and sites at about half of the areas released. and the internal larval parasitoid Tetrasticus um, planopinaceae, which I will just call TETs and OBS in the future here for brevity, again was highly successful. And in our particular plots in Michigan, these are the species whose effect we have been following. The Spatheus agrile external larval parasitoid essentially failed to establish except in a smattering of cases, and attention to this species has shifted away and is now focused on kind of an ecologically similar species, Spatheus galeni from Russia, that we hope will prove more successful in the future. So at this junction, an important question to address is, will these introduced Asian parasitoids be relatively safe to the North American insect fauna? We wish to lower the density of EAB to protect the ash trees, the ash community, and the insects that depend on ash, but we wish not to cause population level damage to related native borers, particularly those most similar in the genus Agrilis. Well, the answer to that question is really found in the host range testing data that is filed at the time of petition for release. And I'm just going to run through that briefly for the 3 Chinese species. And the way this is done is one first starts by looking at other boring insects that are fairly dissimilar to the host, EAB, to see whether they might be quite wide in their host range. So one way to do that is to collect wood-boring insects in the native range where the parasitoid being evaluated, in this case Spatheus agrile, is known to occur. And for 17 species of wood-boring insects collected, including 6 species of Agrilis that were collected in good numbers, over 2,000 larvae, there were no observed attacks. The next phase of this sort of program is to bring things into the lab and test species of borers as one can obtain them from the field. There were 7 species of borers tested in this way that were in families other than the buprestids, and there was no attack. And there was, at this phase, 1 species of Agrilis, which was also not attacked. This was later expanded in the US and China, and at that stage we found that this species does attack in the laboratory certain other Agrilis species when presented in a small container. But other species were not attacked. And then a further factor that has relevance, particularly to this species, is that it was found in tests in a machine that measures response to odors, that this parasitoid is attracted to its host based on smelling ash foliage and will move towards those odors, but not move towards the odors from other ash trees or other kinds of trees different from ash. This then acts as a filter that allows this parasitoid's actions to be pretty well focused on emerald ash borer, and for that reason it was permitted to be released. For the egg parasitoid, more or less kind of the same system. For non-beetle eggs, there was no attack. For beetle eggs in families different from brutestids, there was no attack. For other buprestids in the genus Agrilis, there was attack if they were in the lab and they were similar in size to the EAB. So some pest species like bronze birch borer, two-line chestnut borer, and redneck cane borer were attacked, but species that were more different in size were not attacked. And when you gave them a choice, they preferred EAB. And so based on this information, APHIS judged that on balance this was safe enough, given the level of damage that was occurring, to be permitted for release. The internal larval parasitoid, the TETs, proved to be the most specific of the parasitoids being evaluated from China. There were no attacks on insects in other orders or other borer families, nor were there attacks on 5 other species of Agrylus or 3 other species of Buprestids in a different genus, Chrysobothris. And so based on that, again, it was deemed to be safe enough for release. So the next phase of activity then was to try to measure in the field whether these species can have any impact on EAB densities and whether that promotes survival of ash trees. So we set up research plots in cooperation with Leah Bauer's lab, who's located at the Michigan State University, about right in there somewhere. And we initially set up 3 pairs of release and control plots in 2008, and then added 3 more in 2010. And in 2009 and 10, we made releases of all 3 species of parasitoids in those plots, and then stopped with releases. And now I'm going to go through and sort of give you a history of what we found in those plots in our annual censusing. So every year we go out to all 12 plots, and we cut down 4 trees in each plot, and we completely census it for EAB larvae. And then to detect EAB egg parasitism, we have to find eggs, which are not under the bark, but are rather not where the larvae are under the bark, but rather just in the bark flakes. So either one stands next to trees in the field and uses a pocket knife, as you can see here, to flake off bits of bark and find eggs and then score them for parasitism or not. Or you can use a draw knife to scrape bark down into a plastic sheet, return it to the lab, and sort through that material to find eggs to score them for parasitism. For the larval parasitoids, such as the Tetrasticus planopineaceae, that you can see in this larva here, you can see its immature larva, like there and there and there and there, like sausages inside a semi-translucent sock. And you might see that in the field, or you might see larvae that have broken out of the dead larvae. And the way you find those things is to take the bark off a tree with a draw knife and meter by meter work your way up the tree, dissecting it and scoring everything you find, noting all the instances of parasitism and all other EAB larvae and what they died of. And so these are some pictures of some of the kinds of deaths that might occur to EAB larvae that we score in that sampling process. For example, here's a larva that when it got up here got killed by the tree through some combination of callus growth and necrotic growth around, or development of necrotic tissue around the larvae, killing it. Here's a larvae that died from disease in its burrow. Here's a woodpecker, and woodpecker attack through the bark is very visible and can be directly linked to individual galleries under the bark, so that we can tell, okay, that particular larva was taken by a woodpecker. Or you might see various sorts of parasitoids in one stage or another as larvae or cocoons, which can be recognized as species. Or if you see none of that, you just find a live larvae, we collect it, take it back to the lab, and cut it open and dissect it to see if it might have some very early stages of parasitism. And so for the larval the egg parasitoid, what we have found is that for the first several years, egg parasitism in our release plots shown here was very low, less than 5%, but that it has since increased. Now these are a little bit dated, these data in this particular slide, but levels of parasitism have reached about the 20% level and have stayed that. Now, the difference between the blue and the green are two different sampling methods. This is the bark sifting method, which seems to be more effective at detecting parasitized eggs. We also found that in parasitism has spread after a number of years to nearby control plots where it was not released. This is a very, very small parasitoid, and it doesn't fly readily. It flies a little bit, jumps around on trees. It tends to be highly localized on individual trees, and so rates of parasitism found by just selecting trees at random, which is all we can do, is influenced by that patchy distribution. But year by year, the percentage of trees in a stand on which the parasite seems to have colonized has been creeping up to about 20% now, and we expect that to continue. For the internal larval parasitoid Tetrasticus planopineae, we saw a sort of similar pattern that we went for 3 years during the period of release and shortly thereafter, where we scarcely saw more than about 5% parasitism. It then, by 2012, began to increase and has now reached the level of probably something on the order of about 25% in our sample trees. And I should mention that our sample tree universe is trees that are about 3 to 5 inches in diameter. We can't work with larger trees, partly because they're too difficult and dangerous to cut down in the field, and also because there aren't very many of them left in our plots. So we look for trees that are in that size class, that are alive, that show signs of current EAB activity, so that we have something to sample. And data from 2015 suggests that this level of tet parasitism seems to be a bit lower, but we don't have the actual numbers yet. And the other thing that we saw in our plots was that over the years, we have seen a steady drop in our measure of EAB abundance. Now, I need to be very particular about describing how we measure EAB abundance. I've just described to you what kinds of trees we select for sampling, and we count what is inside those trees, and then we put it on a unit area basis. There's a formula for calculating the number of square meters of bark that a tree has given its height and its diameter. And we have seen that measure drop steadily from a high in 2009 of 40 to 50 live larvae per square meter to perhaps about 5. Now, a square meter of bark is not just a meter of tree length, because their trees are not a meter around in circumference. So it, depending on the size of the tree, might be 3 to 5 meters length. So whereas the first 15 feet of a tree, say, might have 50 larvae in it in 2009, now it will have about 5 larvae in it. And trees die when they get girdled by burrows, the tunnels of the larvae lining up to completely encircle the tree and cut off the phloem. And as the density goes lower and lower, the probability of that happening is decreasing. Another way we can think about what's going on with EAB is we can use an approach called life tables to calculate what's the rate of growth, because that's what we're interested. Population density and ability to reproduce quickly is what makes a pest a pest. And when we first started our plots in 2009, EAB was multiplying at a rate of about 17-fold a year. And that means that a female one year would leave 17 females the next year. So that's just an explosive rate of growth. Within a year, that had dropped substantially to maybe threefold, which is still a strongly expanding population, but no longer exploding. What we're trying to get to are rates similar to what occur in Asia. Now, in Asia, in susceptible North American trees, we have a rate that's somewhere between 1 and 2. Now, 1 is the magic number in population growth rate. If each individual if one female leaves one female, that's a growth rate of 1, which means no increase in numbers. On North American susceptible ash, it's a bit higher than that. On Asian ash, both of these in Asia, it's below that. So we're currently probably at about 2, maybe a little below 2, and we continue to track that number. So the question then of real interest is, what does all this mean for ash trees? And there are several questions that are involved in this, and we do not have the answers yet, but our plots in Michigan are an excellent tool for letting us find the answers as nature reveals them to us. At the end of the day, trees grow slowly, and to answer the question of will there be ash trees in the future is a question that's going to be answered slowly over the next 10 to 20 years, realistically. But each year we get further insights. So we're very focused on this question of how low must emerald ash borer density go before EAB no longer restrains the survivability of ash as they move through their life stages. Now, as they move through their life stages, they will be susceptible to EAB to varying degrees, and parasites will be able to access the larvae in these trees to varying degrees. So ash do not have much of a seed bank. They last a year or a year and a half, and so in our plots we saw a huge flush of young trees as the old trees died. They were initially in size classes that were immune to EAB attack, but as they get up to about 1 inch in diameter, they start being attacked by EAB. And we are tracking that, and I'll explain more about that in a minute. And so this is a very central question. Will these new crop of trees enjoy some good survival? Will there be more seeds in the future? Well, one of the things we've learned from our plots is that even though this crash that was well underway as we started is now eliminated most of the ash in the plots, there are remaining trees in the 3 to 7, 8 inch classes, even the occasional larger tree, and they're still pumping out seeds. And another factor is then how will the biocontrol efficacy change with tree size? Well, we know something about this already. We know that our tets, which is our best parasitoid to date, can only reach larvae inside trees that are up to about 6 inches in diameter. So that's about here, about 12 centimeters. And beyond that, the tree is just too big for the size of the tube called the ovipositor that these wasps use to get through the bark into the larvae, which is why we're interested in this longer ovipositored species, Spatheus galeni, that I'll mention more about in a minute. So another important question was, will we see the same progression of events that we saw in Michigan? In Michigan, we started parasitoid releases when the densities of EAB in plots were already very, very high. So one might ask, will it help to reduce tree mortality rates if we get parasitoids into a newly infested area early? I think the answer is yes, but the catch is it's hard to observe infestations in their early phase because our trapping techniques are rather poor, and early infestations often occur high in the trees in branches. So in New York, where APHIS started work in 2011, so far it has been a similar pattern as in Michigan, with big trees dying rather quickly, even though parasitoids were being released somewhat earlier in Michigan. We're now working in Connecticut, Massachusetts, and Rhode Island in plots that are at the time of parasitoid release in an earlier phase to further explore this question. As trees age, they will have thicker bark, and that is why this new parasitoid with the longer ovipositor is of great interest to us. Releases of that parasitoid will begin next year on a larger scale, now that the mass rearing of that species is more fully developed. And we just got approval for that this year. So ash regeneration is happening, and we're monitoring what's happening in those saplings. We collect 10 trees from each of our 12 plots in the winter of each year and debark them. We're now into our third year of doing that, and I just wanted to point out the data that we got in the second year, which was the only one we have completed, or the third year is not done. And out of that, a quarter of those trees showed current year attack by EAB. And in those new galleys, there were 116 larvae, so about 1 larvae per sapling. And of those, about 3/4 were too small to have experienced parasitism yet. So we can't learn much from those. But of the quarter that were big enough to have passed their period of exposure to parasites, fully half of them were being attacked. And nearly all of that attack was by this introduced parasitoid, Tetrasticus planopineaceae. So this is hugely important evidence that these introduced parasitoids are going to be providing protection, particularly in this phase of ash tree life. Now we need more data, and that is going on. And as trees do get bigger, not so much in these size classes, but in these size classes, it will be parasites like these and woodpeckers that will be providing more effect. And there's also an effect called the herd effect, familiar to people who work in human inoculation vaccination programs, that if you can drive the population of the pathogen, or in this case, the emerald ash borer, low enough, even unvaccinated, or in this case, unprotected size classes of trees benefit because of that lower density. So I'm going to conclude my talk now to just expand my focus a little bit more on, can biocontrol provide protection, to a look at what is at risk. So we've been talking mostly about, can we protect the ash trees themselves? More than that, there are also very distinctive communities formed by ash trees. In the east, we have, for example, the swamp forest formed by black ash. If black ash disappears, those communities will become marshes of some sort that are not forest stands, because there's really no other tree but black ash that can form that community. In the west, there are bushy sorts of ash trees that are local range species that are in some cases even rare or endangered, that have unique things associated with them in these mountain arroyo communities. We know nothing yet about how emerald ash borer will affect those kinds of situations. Personally, I'm from Oregon, and I'm quite familiar with Oregon white ash, which is a lovely tree growing in swamps. Again, something that EAB is going to affect in the future. And not only will those trees and their communities be affected, but the insects that feed on those trees in a specialized way themselves may be endangered. And Dave Wagner of the Yukon in Connecticut has done a very elegant job of pointing us towards which species of insects depend critically on ash. So this is a major ecological adjustment that's going on. We don't have all the answers yet, but we're beginning to see the outlines. And if you would like more information on any of these aspects that I've spoken with you about today, there is a new free book from the Forest Service on the biology and control of emerald ash borer that was produced last year that, while is maybe 1 year out of date now, summarizes everything that we know about this system and where we think it's going. And you can get that book by communicating with Dick Reardon at this email address requesting for your copy. So with that, Robin, I'll turn the screen back to you and questions. Thank you very much.
Thank you, Roy. That's very interesting. And also, I just wanted to comment that parasitoid sampling looks extremely labor intensive. That's a lot of work. My hat's off to all you folks that have to do that. Let's see, we do have one question. One of our participants joined a little late, but he wanted to know what plans are there to introduce biocontrol parasitoids in other states like the Northeast and Mid-Atlantic, etc.
Well, as I showed on, let me see. Is this still visible to people?
Yes, it is.
Okay. Where's that slide? Here it is. So this map shows releases that have been made, and you can see that releases have been made, for example, in North Carolina, Virginia, Maryland, West Virginia, Kentucky, Tennessee, Connecticut, Massachusetts, New York, New Hampshire. So those things are all unfolding. Now, one dot means one release, and of course, a state like North Carolina is a big state, and one release certainly does not cover it. But this is very much an ongoing effort, and we would expect many releases to continue to be made for the foreseeable future as this pest spreads and new states need to be covered. I know for example, that releases were just made this year in northern Louisiana, a very newly invaded state.
Okay, let's see, we've got more questions here. There's one question here. I thought that Spatheus release was limited to below 54 degrees. Still the situation?
The initial releases of Spatheus occurred pretty much through the lake states at the time. That was the core of the infestation. As results came in showing that it was not establishing well there, people began to garner ideas about why it wasn't establishing. And there were two ideas that sort of were most common. One was that it might not be adapted so well to the climate there. And the other was that it might come out at a time when the larger larvae that it needed to lay its eggs in weren't available. So as you go north, EAB can have a 2-year life cycle rather than a 1-year life cycle. This can also happen if EAB is attacking a relatively healthy tree, even at lower latitudes. And when it switches to a 2-year life cycle, instead of having large larvae at the end of the year, they may be more present at the early part of the following year, which can disrupt the phenology, the link of timing between parasitoids and their host. And so the thought was to make the releases of Spatheus agrile at more southerly states and see how it did well there, or how well it would do there. And that is the policy currently. That work is going on. We still have not seen, as far as I'm aware, a place where it has really taken off the way Tetrasticus did in the Lake States and the Northeast. So we don't know if we understand the reason for its lack of efficacy or not. But that's what has spurred the interest in this alternative to it, the Spatheus galeni.
All right. Can biocontrol be introduced in western urban ash successfully? What kind of sites would be suitable in areas where there are not extensive ash forests?
I heard western urban ash.
Correct. It says, can biocontrol be introduced in western urban ash successfully?
Well, not at this point in time because the host isn't there. But if you mean if Oregon were invaded by emerald ash borer, would we then go ahead and release parasitoids? I would imagine the answer is yes. How common is ash as a street tree in Oregon? I'm embarrassed to say I'm from Oregon. I'm trying to think. You know, Oregonians aren't big on shade in the summertime the way people are in the East. They've had so much rain through the winter that by the time the summer comes, they're not keen on having so many shade trees. So I guess it would just be a matter of finding communities where there was a decent planting of ash trees. But there are certainly many naturally occurring white ash stands in the Willamette Valley and other parts of Oregon. So wherever emerald ash borer is doing well, we should also probably try to make these introductions, but it would have to be tailored to the location, and that will vary as the western part of the United States is colonized.
Thank you. We have another question. Your results are quite promising for EAB and ash. Is there any evidence to suggest that biocontrol would be helpful for other borers?
Well, the kind of work that we're talking about, classical biological control, is only relevant for invasive species. So the work would not be relevant for native borers like bronze birch borer, for example, that periodically do become high density outbreak species under some circumstances, but generally are not. So not for native borers, but if we have invasive borers, the answer could be possibly, but again, there are many factors that can complicate it. So we do have a similar situation going on in California where something called gold-spotted oak borer has invaded California from part of its native range in Arizona, and in California is now killing wonderful species of oak, like California live oak and canyon oak. And there is an interest in biological control of it. So far, that has not been productive. These oak trees, for one thing, have enormously thick bark as a development, I suppose, for fire protection. And it's been not the case that they seem to have a good, well-developed group of larval parasitoids that attack through the bark. I mean, the bark on these trees is literally inches thick. So yes, in principle, but the devil's in the details.
All right, thank you. Here's another question is, are you optimistic about the future of North American ash species? In a conversation with Deb McCullough, her opinion was not very optimistic for biocontrol of EAB simply because there was no history of success in biocontrol of introduced buprestid species in the past. Would you say we might witness the first successful biocontrol of a buprestid?
Yes, I would say that. Deb is absolutely right that there's no precedent for this, but that's largely because no one has ever tried. There wasn't really the need, um, and everything has a first case. And for control of invasive buprestids, EAB is the first case. Will ash resume its former status as a tree par excellence that had rapid growth and was virtually pest free? Maybe not completely, because I suspect that it will always bear the burden of having some level of EAB attack, which will reduce its vigor and reduce the quality of the wood. But can we keep it in the forest as a component of the communities that are there? And will it be around to produce wood and firewood? I hope so. But clearly, these are long term questions that it's foolish to prejudge. I think the sensible thing to do is monitor, 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 and see in detail what actually happens. And that is what we are doing.
Okay, another question here. It seems like a wood-boring insect isn't a very good candidate for effective biocontrol. Are there historical examples or current examples where classical biocontrol has been used to provide control of any wood-boring beetle?
There is just one, as far as I know, and that's for a eucalyptus borer that was controlled with an egg parasitoid. Um, there were also larval parasitoids involved, but it seemed to be the egg parasitoid that suppressed it. This was work of Tim Payne in Riverside, and but the flip side of that is that while there are few examples, there have been almost no attempts. And obviously, one can't have a successful example until one has had an attempt. But my feeling is a wood boring insect, as long as it does not carry a systemic fatal fungal pathogen, has at least some chance of being controlled. I think the fate of ash is going to be determined by a complex mix of the tree's own genetics, some of the native species that already are present that attack this insect, and by the introduced parasitoids. Clearly, this is not as easy a system as, for example, a mealybug or a whitefly, where the insect is fully exposed, and we know for a fact their populations are typically natural enemy limited, and explosions occur when they become separated. We are really breaking new ground here. I believe it's valuable to do so.
All right, that looks like all the questions we have at this point. I have to say, Dr. Van Driesche, we've been very fortunate to have you as a presenter today. You've really answered a lot of questions with your presentation and with your Q&A that we are, a lot of questions that we are asking and have heard from, different sides. People have asked me a lot of that as inquiries on the emeraldashboard.info website. So it's good to be able to say, watch the webinar, you'll get your answers. So that's been great that you've been able to do that for us. I wanted to just make a quick request of everyone, if you could please fill out the survey and for us, and so we can keep these webinars coming. The Forest Service has been very good at trying to keep the funding coming so we can continue to bring you these for free and bring presenters like Dr. Van Driesche. I'm going to be closing the webinar here in just a minute unless there's another question. Hold on here, we might, oh, okay. Tom says, thank you very much, it's been very interesting. Thank you, Tom, for being online with us. And I will be sending out an email, and you'll get also a link to that survey as well. Is there anything else you would like to comment on before we are done, Roy?
I would just stress that these are long-term events, and while we have been working on this now for 10 years or so, um, keep in mind that that's in many areas only about 5 or 6 generations of this insect, since in many areas it takes 2 growing seasons to complete a generation. And these kinds of adjustments clearly take 10 to 20 host generations to occur. And while that may occur in a single year in something as easy as a mealybug, for a borer and a host plant like a tree, we must stay the course and follow out events as they unfold. And I am optimistic, but I do think that they will unfold over a fairly considerable piece of time, and that the important thing is to know what happens. And we only know if we look. And so that's the role of post-release monitoring of a biocontrol program. Thank you very much for having me on the program today.
Thank you, Dr. Van Driesche. I'm going to stop the meeting here, and I want to thank everyone for attending. Have a good day.